Good day, students. Today's practical is titled Petiole Preparation of a Dicot Plant. This is a dicot plant. You can see it has a broad name. And the name of this plant is Alternanthera sessilis. Now, this is a petiole. The petiole is a part of the plant that attach the plant, the leaf, to the stem of the plant. And it's also the part that holds the midrib of the leaf. It attaches the leaf to the stem of the plant and also holds the midrib. This is the midrib of the leaf. Thank you. So now the aim of this experiment is to prepare a cross section of a mature petiole, which is a dicot plant, so you can see the internal structure. Now the materials, we'll be using a dicot plant, using our distilled water with ethanol, with distilled water, petri dish with distilled water, petri dish with sodium hypochlorite. We'll be using different grades of alcohol. We're using Safranin Glycerol, nail polish, our slide and cover stick. Now the procedure. You cut, this is the petiole, you cut the petiole, away from the plant. Let me show you how I cut it again. You cut the petiole away from the plant. So you put the petiole between this styrofoam material so you can form a strong block. So when you are sectioning, it will hold it very tight, so it will be easier to section. So you now cut this section. You are using this styrofoam material to mimic a microtome machine, which is a machine used to make section of a plant tissue. So you come into the distilled water and you make several sections. You must make sure you make a very thin section so that light of the microscope can penetrate when you are viewing. So you make series of sections. You make sections, series of cut sections into the water. You make series of cut sections. You just from right to the left. You just cut. Transfer inside the water. So you cut. You might find the styrofoam material inside your water. You just remove it, and being a whitish substance, it will not affect, it won't interfere with our. So after you have made this section, we section inside water to hydrate the cell. So you pick one of those sections and put in the cover slide or on a glass slide. You put one of these sections in a glass slide, and then to hydrate the cell, you now put a drop of sodium hypochlorite to decolorize the cell. Leave it for like 10 seconds. Sodium hypochlorite to decolorize the cell for about 10 seconds. So you remove the effect, and you add also a drop of distilled water again to remove the effect of the decolorizer. After which we have hydrated the cell. Now we want to decolorize the cell for proper and accurate staining. So to decolorize the cell, we do it in grades of alcohol. 50% first, a drop of 50% alcohol for just 5 seconds. Then you put 70% alcohol. We are dehydrating the cell gradually to prevent breakage of the cell. Then you also put 100% alcohol. Then you can drain the excess. You can drain the alcohol. Your sample must be at the middle of your slide. And the sample that you are preparing must always be at the middle of your slide. Then you put a drop of Safranin to stain the cells and allow it to stay for 30 seconds. After 30 seconds, you put a drop of glycerol, or you can also put 2 drops. The glycerol does not really have, you can put 1 to 2 drops, and then you cover gently from one side with cover slip. This glycerol will enhance easy visibility. This glycerol that we put will enhance what? Easy visibility of your sample. Then as you are checking under the microscope, so we now will cover slip from one side to avoid trapping of air bubbles. So we gently lower it, and then we make sure that the cover slip contains our stain with the stain sample. So we drain the excess stain with our tissue. We wipe the base so that we will not stain our microscope. So we now seal the edge of this preparation with nail varnish. That is, we seal the edge with nail varnish to prevent the hydration of our prepared sample. And this prepared sample can stay for many weeks to few months, from 1 week to about 6 months or almost a year, and it will still remain intact if properly kept. Thank you. Thank you for watching. If you find this video useful, please kindly like and share and subscribe. God bless you.